Hello, good morning students. Welcome to the Unacademy special YouTube class sessions. So welcome to all the students to this platform. So warm welcome to each and everyone. So once again welcome to the YouTube special Unacademy classes. Students, before going to know this, as you all know, Unacademy has brought out with the Plus subscription. So in this you can have a premium content at affordable price, structured live courses with dedicated live doubt clearing sessions, quizzes at a regular intervals of time for self-analysis, batches for complete preparation, test series for DPPs, full-length marks with analysis. So if you get a single subscription, so you will get unlimited access. So for that you can use the code for getting 10% instant off is GHOUSELIVE. So once again, warm welcome to all the students to this special YouTube sessions. So today our discussion for the topic of the day is we are going to discuss how the chromosomes, what are the different types of chromosomes are there, and we are going to study about that. Students, those who are joining right now, warm welcome to each and everyone. Warm welcome to each and everyone. So good morning all. Good morning all. So let me start with the nucleus, students, because before going to learn or before going to know about the types of chromosomes, our first thing is we need to know how the structure of nucleus is. Then we can go for the chromosome structures, students. Okay, so today our topic for the YouTube session is types of chromosomes. In this we are going to start nucleus. Student, here nucleus as a cell organelle was first described by the scientist Robert Brown. So the Robert Brown is a scientist. He described the nucleus as cell organelle in the year of 1831. In the year of 1831. So students, you can have the picture of Robert Brown on your screen. So he is the Robert Brown. So he has told about the nucleus as a cell organelles for everyone. Student, here once you observe the nucleus, you see here guys. So as everyone knows, there is a cell like this. Inside the cell, students, a nucleus will be there. So the nucleus is double membranous. Nucleus is what? Double membranous, not continuous, and leaves a pores called what? Nuclear pores. And leaves a pores called what? Nuclear pores. You can see this. And endoplasmic reticulum is following to that. On the surface of endoplasmic reticulum, you can observe ribosomes on that. So this was the structure of the nucleus first. So now let us study this about the chromosomes. So students, the nucleus was stained by the basic dyes. Nucleus was stained by the basic dyes and was given the name as chromatin by Fleming. Students, this is also one of the another important point you have to keep in your mind. The nucleus was stained by the basic dyes and it was given the name as chromatin. So many people, they'll get confused. What is a chromatin? What is a chromosome? So student, the pre-form of the chromosome is nothing but what? Chromatin. So now chromatin gets condensed, then it converts into chromosome. Students, everyone, you need to keep in mind this. Chromatin is a pre-form of the chromosomes. So that was stained by basic dyes. Basic dyes means, students, so if chromatin gets stained by basic dyes, basic dyes. So now we are using basic dyes means, then what is the nature of the chromatin? Question mark here. Students, you can answer here. What is the nature of chromatin? Students, you can have the picture here. What is the nature of chromatin? Because you are using basic dyes. When you are using the basic dyes, then what is the nature of chromatin? So obviously the nature of chromatin is acidic nature. That's the reason why, students, we are using basic dyes. So this was given by the scientist Fleming. Who given this one to all of us is Fleming. So you can have a picture on the screen. Now students, next you observe here the interphase nucleus. So what is the interphase nucleus? Interphase means what? Resting phase. Interphase means yes, the phase present between two successive M is nothing but interphase. So in this interphase, the common name which we call for interphase is what? Resting phase. We are calling it as resting phase. Interphase is nothing but resting phase. So here you see here, what is resting phase here means? Here you see in the brackets, nucleus of a cell, okay, when it is not dividing. So if a cell is not dividing, if a nucleus is not dividing, then that particular we are calling it as a interphase nucleus. Okay. Now student, in the interphase nucleus, you can observe highly extended and elaborate nucleoprotein fibers. Highly extended and elaborate nucleoprotein fibers called chromatin. You understand now? So in interphase nucleus, you can observe the chromatin. In the normal dividing cells, you can observe chromosomes. In normal dividing cells, what you can observe? Chromosomes. Hope you understand the difference between chromatin as well as the chromosomes. Fine, students. Now here, another important term, you may have a doubt now. Sir, what do you mean by nucleoprotein fibers? So the nature of chromatin is nucleoprotein. What do you mean by nucleoprotein here? Proteins plus nucleic acids. Proteins plus nucleic acids. That nature is called as nucleoprotein. So students, generally you study here glycoprotein. What do you mean by glycoprotein? Then glycolipid. What do you mean by glycolipid? So glycoprotein means carbohydrate plus protein. Glycolipid means what? Carbohydrates plus lipid. So this is a glycolipid, this is a glycoprotein. This is glycolipid, this is a glycoprotein. So like this, students, nucleoprotein means what here? Proteins plus nucleic acid. This is one of the most important point you all has to keep in your minds. You see, students, here the chromatin fibers, how they condensed into nucleus. You can see here from the nucleus, chromatin fiber, DNA, you can see nucleosome. Yes. So then it becomes condensed to chromosomes. Now students, the interphase nucleus having nuclear matrix and one or more spherical bodies called nucleoli or nucleolus. Students, inside the nucleus, because you see this is a cell, inside the cell is a nucleus. Inside the nucleus you can see small spherical bodies. So the small spherical bodies we are calling it as nucleoli. Singular we call it as a nucleolus. Student, this is a membrana, membranous cell organella. Yes, it's just like as ribosome. But ribosome is membraneless, right? No, non-membranous, right? So nucleolus is a non-membranous. Nucleolus is a what, student? Non-membranous. Okay, so it is present inside the nucleus. Now, what is the main function of the nucleolus? You can see, students, what is the nucleolus? Inside the nucleus, you can see. Yes. Now, the main function is what? It produces rRNA. The main function of nucleolus is it produces rRNA. What do you mean by rRNA? rRNA means ribosomal RNA. rRNA means what, student? Ribosomal RNA. Very good, super. Now, students, coming to here, the nuclear envelope consists of two parallel membranes with the space between, yes, what is the space here? 10 to 50 nanometers, that we are calling it as perinuclear space. As I told you in the beginning of my session, nucleus is surrounded by how many membranes? It is surrounded by two membranes. Okay, the gap, okay, or the space present between these two membranes, we are calling it as perinuclear space. And what is its dimension, students? What is its dimensions? Yes, the dimension is 10 to 50 nanometers. The dimension is what, guys? 10 to 50 nanometers. Since check the units here. They may ask in angstroms like this. Okay, you should not confuse, guys. See, the term chromatin was coined by, right? The other question for you on your screen. You can predict the answer and you can share your answers in the comment box too. Students, you can please share your answer for this. Superb. Yes, Fleming. So, very first time, who coined the term chromatin means, so you have to think about the scientist name called Fleming. You have to think about the scientist name called Fleming. So, now students, what is the function of perinuclear space? Yes, the perinuclear space forms a barrier between materials present inside the nucleus and that of cytoplasm. It is acting as a barrier so that it allows the substances from nucleus to cytoplasm, from cytoplasm to the nucleus. That's the reason why we are calling it as perinuclear space forms a barrier between the material present inside the nucleus and that of the cytoplasm You see this, the outer membranes of nucleus remains continuous with RER. Yes, the outer membrane, if you see, this is the outer membrane view, students, of this nucleus. See, it is continuous with the RER. What do you mean by RER? Rough endoplasmic reticulum. Why we call it as a rough endoplasmic reticulum, guys? As you all knows, yes. Okay, why we call it as a rough endoplasmic reticulum, students? As you know, rough endoplasmic reticulum having ribosomes on it. It is having ribosomes on it. Yes. So now here is a question for you on the screen. What is perinuclear space? What is perinuclear space, students? Yes, the space present between two nuclear membranes, right? Super. You please hear. The nuclear envelope consists of two parallel membranes. The nuclear envelope consists of how many parallel membranes? Two parallel membranes with a space between 10 to 50 nanometers, that we are calling it as perinuclear space. Now, coming to the next point, the nuclear envelope is interrupted by minute pores formed by the fusion of its two membranes. Yes, that is called nuclear pores. You see some pores on the screen, how nucleus forms pores, right? Super. Now, these nuclear pores are the passages through which movement of RNA and protein molecules takes place in both the directions. So, nuclear pores are the passages, okay, through which the movement of RNA and protein molecules takes place. Between cytoplasm and nucleus. Students, you can ask your doubts in the comment box, guys. Yes, super. Normally, how many nucleus will be there per cell? One per cell. So sometimes it will be more. When it's a more, you can call it as yes, polynucleated. You know that, students. Then another important point I told you already. What is the function of nucleolus? So as you know, the function of nucleolus is active ribosomal RNA synthesis. Then anucleated means what? They do not have the nucleus. Then example, erythrocyte of mammals and seal tube cells of vascular plants. Students, this slide is very important for all of you guys because they'll ask you the examples, okay, which are having nucleus and which do not have the nucleus. So which are having the nucleus, which don't have a nucleus also, So on your screen, which is there? Yes, on the screen you have a erythrocyte of mammals and seal tube cells of vascular plants. So now here is a question again for you. What is a nuclear pore? As I mentioned earlier, the nuclear envelope is interrupted by the minute pores and which are formed by fusion of its two membranes. You see students, RBC, they don't have a nucleus. Sieve tube cells do not have a nucleus. Okay, you see RBC. So RBC is anucleated, right? RBC is anucleated, and whereas sieve tube cells, you see students, you don't have any nucleus. This is the companion cells. So companion cells have a nucleus, not sieve tube. Okay, this is very important, you have to keep in mind. So the pressure gradient in sieve tube cells will be controlled by the nucleus of companion cells. Okay, that you already studied. Now, so students, larger and more numerous nuclei are present in a cell, so which generally carry out protein synthesis. But during different stages of cell division, cells show structured chromosomes in place of nucleus, right? Yes, students, you can see the picture of nucleus on the screen. Hope you all understand. So before going to know about the chromosomes, it is a prerequisite to all of us to know what is a nucleus, right, students? So during dividing state, you can observe the nucleus. So you see students here. You see students, a single human cell has approximately 2-meter thread, is nothing but 23 pairs of chromosome we have. Each chromosome has a primary construction. You can see each chromosome has a primary construction. So each chromosome has a primary construction that is called a centromere, having two chromatids called sister chromatids. You can see clearly on the screen. So what is a kinetochore? On the centromere, disc-shaped structures are present, which are called as kinetochores. So what is the function of kinetochore? The function of kinetochore is what? Yes, to which spindle fibers will bind it. Okay, during cell division, spindle fibers will be attached to the chromosomes. Yes, the point where spindle fibers attached to the chromosome is kinetochore. So I would like to tell once again to all of you students, what is kinetochore means. On the centromere, disc-shaped structures are present. Which are calling it as kinetochores. Now, based on the position of the centromere, the chromosomes can be classified into how many types? 4 types. Chromosomes can be classified into how many types, students? 4 types. One is metacentric, second one is submetacentric, third one is telocentric, and the fourth one is acrocentric. Students, we will study about each and everything now in this YouTube session. You can see here there are 4 types of chromosomes are there. One is called metacentric, the second one is called submetacentric, third one is called telocentric, and the fourth one is called acrocentric. So now students, there is a question for you on the screen. You can just check it, the question here please. So disc-shaped structures on centromeres are called as yes, the answer is what, students? Yes, kinetochores. The answer is kinetochores. I already know about that. Now let me talk about the first form of chromosome, nothing but metacentric, students. Metacentric chromosome has middle centromere forming 2 equal arms of chromosomes. The metacentric chromosome has middle centromere. See here, students, how this is there. So what is this? Middle centromere. This is middle centromere. So centromere is also called as primary construction. Centromere is also called as what, guys? Primary construction. Centromere is also called as primary construction. So the metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. You see here, both are equal arms here. Yes, students. So that type of chromosome, we are referring it as metacentric. Now coming to submetacentric, students, you see here, in submetacentric, chromosome has centromere nearer to one end of the chromosome. I repeat again, in submetacentric chromosome, okay, the chromosome has centromere nearer to one end of the chromosome. So if the centromere is nearer to one end of the chromosome, it results into one shorter arm and one longer arm. It results into one shorter arm and one longer arm. Students, here you can observe very carefully what is submetacentric chromosome. So you have to get a question to yourself. What is submetacentric means? Our arms are equal or unequal? Definitely is unequal. Because why it is unequal? You can have a question here. If you think why it is unequal, because centromere is nearer to one end of the chromosome. Centromere is nearer to one end of the chromosome, resulting into one shorter arm and one longer arm. You see here. This is a longer arm. See here, one is a longer arm. This is a shorter arm. You see, check the previous slide here. In metacentric, students, in metacentric, arms are equal, right? In metacentric, arms are equal. Check once, everyone. Check everyone, please. Yes, arms are equal. But here, students, please check. But here, shorter and longer arm. So what is the black color structure? It is centromere. Students, hope you are understanding the concept better. And you have a doubt, you please share in your comment box. Waiting for your doubts, students, please. You can raise in your comment box. Now, student, the third one, acrocentric. In acrocentric chromosome, the centromere is situated close to its end. So, in metacentric equal, in submetacentric, centromere is very close to one end, right? Nearer to one end. But here, students, it is very close to its end. You can see in acrocentric. Because of that, one is extremely short. One is extremely short, and the another one is very long arm. And the another one is what, students? Very long arm. And the other one is very long arm. You can see, students, in submetacentric also unequal arms. But extremely short arms, like extremely short and extremely long, they are present in which form of chromosomes? They are present in acrocentric form of chromosome. They are presenting in, yes, hope all you are seeing this. Check it here once, students. Acrocentric. You see this very small. This is very small, yes. This is very small. This is very long, as I already mentioned. So you please check this. You please check this. In submetacentric, shorter arm, longer arm, but not extreme, right? You see submetacentric, students. In metacentric, you see both are equal arm. See here, what you have to understand is, if centromere is at exact middle, it results into the equal arms of the chromosome. If the centromere is at middle and arms are equal, then that type of chromosome we are calling it as metacentric. Second one, you see, in submetacentric chromosome, centromere is nearer to one end of the chromosome. Resulting into one shorter arm and one longer arm. That is submetacentric. But extremely short and extremely long, that is acrocentric. Student, you can see here, in acrocentric chromosome, the centromere is situated close to its end, then forming one extremely short and one very long. One extremely short and one very long. You can see this, students. Super. Now, let me go with the last one. So the telocentric chromosome has terminal centromere. Terminal centromere. So you see, student, generally chromosome has two chromatids, right? Okay, suppose this is a chromosome, centromere and one chromatid. This is normal chromosome. Okay. Then here, students, in telocentric only one chromatid is there. Okay, another chromatid is absent. Why? Because it has terminal centromere. This is called as terminal centromere. This is called as terminal centromere. So in the earlier one, it is extremely short and extremely long. But here, yes, students. Okay, fine. Now, let us discuss. Some chromosomes have non-staining secondary constructions at a constant location. Non-staining secondary constructions at a constant location. This gives the appearance of a small fragment called satellite. Student, you see, this is the full structure of the chromosome. You see, this is called centromere. Hope you are understanding the concept better, students. There is nothing but primary construction. So these are the chromatids, students. So these are two sister chromatids, right? Okay, this is chromatids. You see here, some chromosomes have non-staining. So these are non-staining. Means you can't stain. Non-staining. Okay, this satellite is there, no? This is called a satellite. This satellite is nothing but secondary constructions. So primary construction is, students, and secondary construction also, what is primary construction and what is secondary construction. You have to understand. The primary construction is centromere, and whereas secondary construction is satellite DNA. Yes. Now chromatin appears on bead salt string. So the beads are known as nucleosomes. Yes, students, today our topic is types of chromosomes. You can see, guys, once again I will show you this, all the slides. Once again you can see how many chromosomes which have in our body. 23 pairs. Every chromosome has a primary construction, that is called a centromere. So on this centromere, disc shaped structures are present which are called as kinetochores. So based on position of centromere, the chromosomes can be classified into 4 types: metacentric, submetacentric, telo, and acrocentric. So disc-shaped proteins called as a kinetochores. Student, metacentric, first of all, metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. Same in submetacentric, you can observe near to one end, results in very short and very long. Submetacentric. Coming to acrocentric chromosome, centromere is situated close to its end, then forming one extremely short and one very long arm. That is acrocentric. Then telocentric, you can see it has a terminal centromere. Then you see, students, some chromosomes have non-staining secondary constructions at a constant locations that are called as a satellite DNA. So they are non-stainable, means you cannot stain that. Okay, you can see here also. This is also satellite DNA. You can see in the picture also, students, this is satellite DNA. So the chromatin beads, beads all string. So that beads are known as nucleosomes. The beads are known as nucleosomes. Hope, student, fine. So I welcome you all to ask your doubts in your YouTube comment box session. You please raise your questions in your comment box, students. You please raise your questions in comment box. You please ask your doubts, students. You ask your doubts, please. So hope today class we have gone through the clear presentation of types of chromosomes. So not only the types of chromosomes, students, we have studied what is a nucleus and what is the structure of nucleus, what is nucleolus and what is the structure of nucleolus and what is the function of nucleolus. Then we studied types of chromosomes. There are 4 types of chromosomes: metacentric, submetacentric, acrocentric and telocentric. Remember, telocentric chromosome has only one chromatid because yes, centromere is at tip. Okay guys, so thank you so much for attending this special YouTube session. So once again, I would like to thank all the students who attended special YouTube classes.